Biochemistry 401 Lecture 5. Today we're going to talk about DNA, RNA, and protein. We're going to start with the central dogma. We'll talk about the rules of the central dogma and then the exceptions. We're then going to talk about the structure of DNA and go on to an overview of replication, transcription, and translation, and we'll finish with the genetic code. The central dogma was first proposed in the middle of the 20th century by Watson and Crick, and it essentially says this: genetic information that gives individuals their unique characteristics is stored in DNA molecules. This genetic information is passed on from cell to cell and from parent to progeny. But before it can be passed on, this DNA must be replicated, copied, through a process called replication. During replication, DNA is used as a template for the synthesis of new DNA strands. DNA can also be used as a template for the synthesis of RNA in a process called transcription. And again, the language is nucleotides, in this case, ribonucleotides. And finally, the central dogma says that RNA can be used as a template for the synthesis of protein. And instead of nucleic acids, this language is written in amino acids. And so, since we're going from a language of nucleotides to one of amino acids, we're translating from one language to another, and this process is called translation. But there are exceptions. RNA can also be used as a template for the synthesis of DNA. This happens through a process called reverse transcription. This occurs in retroviruses that have an RNA genome and must synthesize DNA using an RNA template. This DNA can then be inserted into the host genome, or it can be used to transcribe retroviral genes. Now, in our cells, we also have reverse transcription occurring. This is because the chromosomes of eukaryotes are linear, and at the ends of these chromosomes are sections of DNA called telomeres. During repeated replications, these telomeres can become shortened, and so, in an effort to prevent this, the enzyme telomerase uses an RNA template to synthesize added lengths of DNA to extend the telomeres. Nucleic acids, like DNA and RNA, are comprised of nucleotides, and nucleotides themselves are comprised of three things: one or more phosphate groups, a nitrogenous base, and a pentose sugar, similar to the one that's shown here. Now, it's important to understand the numbering system for pentose sugars and nucleotides, so let's go over that. First of all, we're going to start with carbon number 1. And we know that this is carbon number 1 because it's the only carbon that is bonded to two oxygen atoms at the same time. This 1' hydroxyl is going to be the site of attachment for nitrogenous bases, and we'll see that in a minute. Now, the 2' position is also very important. This contains the variable group, either an OH for ribose or a hydrogen for deoxyribose. The 3' position is also very important. This is going to be the site of attachment between one nucleotide and the next one in line. This is going to happen through nucleophilic attack by the 3' hydroxyl on a phosphate group of the incoming nucleotide, and we'll see that in a minute too. Next comes the 4' position, and finally the 5' carbon. Attached to the 5' carbon is the 5' hydroxyl. This is also very important, because this is going to be the site of attachment for one or more phosphates. Now, the reason that the numbering in this sugar is given a prime designation is to distinguish them between carbons in the nitrogenous bases. Those do not have prime designations, whereas the carbons in the sugar do. So now let's talk about the difference between a nucleoside and a nucleotide. We're going to first attach a base to the 1' position. This is now a nucleoside. It's comprised of a pentose sugar, either ribose or deoxyribose, and a nitrogenous base. A nucleotide has a phosphate attached to the 5' hydroxyl. Nucleotides can have 1, 2, or 3 phosphates. Here is adenosine triphosphate, ATP. This is an RNA nucleoside with 3 phosphate groups, alpha, then beta, and finally gamma is the last one. It's important to remember this. The nitrogenous bases in DNA and RNA are shown here. They consist of 2 types, based on whether they have 2 rings or only 1 ring. Purines have 2 rings, and they include adenine and guanine. Pyrimidines have 1 ring, and they include cytosine in both DNA and RNA, and in RNA only, uracil, and in DNA only, thymine. And so DNA and RNA both contain adenine, guanine, and cytosine. RNA contains uracil, and DNA contains thymine as well. It's important to remember this. These nitrogenous bases form base pairs through hydrogen bonding. Guanine and cytosine in both DNA and RNA can form 3 hydrogen bonds. 2 hydrogen bonds are formed between adenine and thymine in DNA and adenine and uracil in RNA. Because of the differences in bonding patterns, the strength of the base pairs is different between guanine and cytosine, adenine and thymine, and adenine and uracil. Guanine and cytosine is the strongest, adenine and thymine is the second strongest, and adenine and uracil are the weakest. Now it's thought that RNA was the first nucleic acid that was built, and so a long time ago, before there were even cells, there was RNA, it's thought. It is also thought that the earliest forms of life had an RNA genome. Over time, organisms that had a different type of genome, a DNA genome, had a distinct advantage, and they won out. This genome must be kept safe because it is the genetic information that's passed down from cell to cell and from parent to offspring through the generations. Now, while some mutations are beneficial, this is how organisms evolve, other mutations can be very devastating. DNA is more resistant to mutation than RNA. And this is because DNA has a 2' hydrogen rather than a 2' hydroxyl as is seen in RNA. RNA is especially prone to hydrolysis because of the 2' hydroxyl. So how does this hydrolysis happen? Well, we start off with acid-base chemistry. A hydroxide is going to come along and pull a proton off the 2' hydroxyl, which causes it to become a nucleophile and attack the phosphate backbone. This results in a breaking of the phosphate backbone, but another phosphodiester bond is made. Instead, this is a 2' 3' phosphodiester bond, resulting in a cyclic molecule. This is a highly strained cyclic structure, and it resolves to either a nucleotide 3-phosphate or a nucleotide 2-phosphate. And that's hydrolysis. And it's all because the 2' hydroxyl is a more reactive group than a hydrogen. And so DNA is a more stable molecule. In fact, a fragment of the leg bone of an ancient ancestor to the horse was dug up from the Canadian permafrost. This bone fragment was determined to be anywhere from 560,000 to 780,000 years old. This DNA was in such good condition that scientists were able to reconstruct the genome of this ancient horse. Wow. This is pretty stable. In this figure, we see Przewalski's horse. This is the last truly wild horse in existence. We use RNA as working copies, so in many cases we can use this information to make protein. Now, you don't necessarily want these working copies to be in existence for a long time, because sometimes you want to make a certain protein at a specific time, at a specific place, and then you don't want that anymore. You want to stop making that. And so if you want to stop making the protein, You have to find a way to stop making this working copy available for synthesis. And that's usually through hydrolysis. So, DNA stability, excellent for genomes. RNA instability, excellent for working copies. DNA is a double-stranded helix where one strand winds around the other in an anti-parallel fashion, with one strand going in one direction, 5' to 3', and the other strand going in the opposite direction. These strands bind together to one another, predominantly through complementary base pairing between nitrogenous bases. In this figure on the left, we see DNA that's in a ball and stick diagram with individual atoms colored according to the elements that comprise them. On the right side, we see a space-filling diagram. The sugar phosphate backbone is shown in red. One strand is lighter than the other, and the bases are shown in blue and gray. One strand has a darker phosphate backbone and blue nitrogenous bases, and the other strand has a lighter shade of red for the sugar phosphate backbone, and the nitrogenous bases are shown in gray. The bases stack together in the center of the DNA molecule, and the phosphate backbone runs along the outside. Some of the associations that allow this structure to have such stability, again, certainly are the hydrogen bonds between the nitrogenous bases, but also there's a component of hydrophobic interaction caused by the stacking of these bases one upon another in the center of the molecule. The outer region of the DNA is highly negatively charged because of the phosphate groups in the DNA backbone. Replication is semi-conservative. The resultant DNA strands that are made consist of one parental strand and one newly synthesized strand. This is a process that requires DNA template, deoxynucleotides, an RNA primer, and an enzyme catalyst, DNA polymerase. Each template strand is read 3' to 5', and its complementary strand is synthesized 5' to 3'. In a process that probably harkens back to the time when RNA was the primary nucleic acid in the genome, RNA is polymerized first as a primer, and then DNA is polymerized. These ribonucleotides must be removed. Then DNA is synthesized to replace these excised ribonucleotides. And finally, any breaks in the DNA backbone are sealed using an enzyme called DNA ligase. This is an enzyme that requires ATP for energy. And so, during replication, the strands separate. RNA is polymerized first as a primer, and then DNA is polymerized. The RNA is removed, DNA replaces the RNA, and then any breaks in the nucleic acid backbone are sealed through the action of ligase. The biochemical mechanism for the addition of nucleotides to a DNA strand during replication is this. A primer has already been added to the DNA template, and an incoming deoxyribonucleotide enters. The 3' hydroxyl makes a nucleophilic attack on the alpha phosphate of the incoming deoxynucleotide, and the gamma and beta phosphates leave as pyrophosphate. This is very important, and we'll talk about that in just a minute. Pyrophosphate is immediately hydrolyzed to 2 molecules of inorganic phosphate, and so at the end of the reaction, we're left with 2 molecules of inorganic phosphate and a DNA strand that's been lengthened by 1 deoxyribonucleotide. And again, this synthesis proceeds from 5' to 3', because during replication, we're adding to the 3' hydroxyl of the previous nucleotide. This reaction is driven forward by the loss of pyrophosphate through hydrolysis to 2 molecules of inorganic phosphate. Through the loss of the product, Le Chatelier's principle comes into play. Molecules of inorganic phosphate have several resonance structures. Because of this, they're at a lower energy state than they would be in a nucleotide triphosphate or in pyrophosphate. And so this helps to decrease the enthalpy of the products compared to the reactants. Also, two molecules of inorganic phosphate can orient themselves randomly in solution. The nucleotide triphosphate's position is now less random, but the two molecules of inorganic phosphate help to make up for that. And all these things contribute to the favorability of this reaction going in the forward direction. And so these replication notes are for you. Replication is the synthesis of DNA from a DNA template and deoxyribonucleotide triphosphates. Replication requires a primer. Generally, this is an RNA primer. Replication is carried out by DNA polymerase. Replication is semi-conservative. Replication generally occurs prior to cell division. One replicated genome can be passed on to the daughter cell, while the other stays in the parent cell. And again, these are more notes for you. Replication has these characteristics. Replication requires a template which is read in the 3' to 5' direction. Synthesized DNA is complementary to the template strand. Replication requires a primer with a free 3' hydroxyl. Replication extends the chain in the 5' to 3' direction. During replication, deoxyribonucleotide triphosphates react through nucleophilic attack by the preceding nucleotide's free 3' hydroxyl on the alpha phosphate of the incoming deoxyribonucleotide triphosphate. This reaction yields one molecule of pyrophosphate and a chain extended by one deoxynucleotide. Pyrophosphate is then quickly hydrolyzed to two molecules of inorganic phosphate, orthophosphate. The polymerase reaction is catalyzed by a class of enzymes that are called DNA polymerases. So now let's talk about transcription. This is making an RNA transcript from a DNA template. It's the process by which RNA is polymerized. This process requires three things: a DNA template, nucleotide triphosphates, and an enzyme called RNA polymerase. The process of transcription also uses a DNA template. The template strand is read 3' to 5', and a complementary RNA strand is synthesized 5' to 3'. The mechanism for ribonucleotide addition to a growing RNA chain is similar to that of DNA, only there's no primer necessary. There is no primer necessary for RNA synthesis, and the nucleotides that are being added are ribonucleotides, not deoxyribonucleotides. The ribonucleotide 3-phosphate will come in, 3' hydroxyl of the previous nucleotide will make a nucleophilic attack, pyrophosphate will leave, and will be immediately hydrolyzed to two molecules of inorganic phosphate, and so you'll be left with an RNA chain that's increased by one ribonucleotide and two molecules of inorganic phosphate. And again, it is this loss of pyrophosphate and the subsequent hydrolysis that helps to drive this reaction forward. The genome is pretty large, and so to start transcription, RNA polymerase needs to know where to start and where to stop. Therefore, to be able to transcribe the message correctly, the polymerase has to find the right stack, the right book, and the right page to make sure that the right transcript is made so that the protein product can be made correctly. So how is RNA polymerase brought to the correct start site to begin transcription? There are regions of the DNA that are designed to promote transcription through attracting the DNA polymerase to the start site. These regions are called promoters, and they're different in bacteria and eukaryotes. In bacteria, promoters are generally comprised of a -35 and -10 region. The -10 region is also called the Pribnow box. So, what does -35 and what does -10 mean? Well, if you consider the spot where the polymerase is actually going to start synthesizing RNA, that first nucleotide that's going to be synthesized is going to be complementary to position number 1. The region just before that is not called 0, it's called -1. So the -10 site is 10 deoxynucleotides away from the start site of transcription. Likewise, the -35 region is 35 deoxynucleotides away from the start site of transcription. In eukaryotes, the promoters are much more complicated. We're more complicated organisms, and it makes sense that we would have more complicated ways of starting transcription. We have to be really careful when we generate RNAs that we're giving the right instructions at the right time. Otherwise, things can go really haywire. So, we have something that's called the TATA box, and you need to remember this name. This is usually about 25 deoxynucleotides away from the start site. Some promoters also contain other sites, including this cat box that's shown here. So you need to know where to start. That's true. You also need to know where to stop. And for this purpose, there are special sequences that identify transcription stop sites. So when RNA is synthesized, it starts to fold up into complex structures through base pairing. Forming interactions generally reduces the enthalpy of the molecule. So if this molecule can make hydrogen bonds between G and C and A and U, it certainly is going to try to do so. And so this molecule has folded up to make a stem comprised mainly of GC base pairs and also an AU base pair in there for good measure. And at the end, there's a loop region where there is no base pairing. This RNA is still attached to the DNA template. However, the formation of this stem loop causes some stress in the molecule. And because of this structure, the RNA polymerase will pause. And to release the tension, something is going to have to give. And it's not going to be those GC bonds. Those are too strong. And so the stem loop structure is not going to be released in order to get rid of this tension. Instead, it's going to be the portion of the RNA that's stuck to the DNA template. Those bonds will be broken, and the RNA will be released from the template. RNA molecules serve many purposes besides synthesizing protein. They're also used for enzymes, as binding partners of RNAs in biological processes, and also as regulatory molecules. The three major types of RNA that are found in E. coli and also in eukaryotes are ribosomal RNA, abbreviated rRNA, and this is the RNA that helps to form the ribosome. And the second type of RNA is transfer RNA. It's abbreviated tRNA. We'll talk about this in a minute. Messenger RNA is the final type, and it's abbreviated mRNA. And this is the type of RNA that's used as a template for protein synthesis. In the world we live in, there are many conventions. One of them has to do with automobiles. If you're driving a standard car, the clutch is on the left, the brake is in the middle, and the gas is on the right, and that's by convention. To mix them up can be a big problem. Likewise, we also have conventions when we're trying to convey information regarding DNA. If there's only one strand written, it's written 5' to 3', and we assume that it's the coding strand of DNA. And portions of this coding strand will have the same sequence as the RNA that's going to be synthesized. If two strands are written, the coding strand is on the top, written 5' to 3', and the template strand, shown here in blue, is written 3' to 5', and this is the template strand from which the RNA will be synthesized. The red nucleotides shown here represent the TATA box. During RNA synthesis, the coding strand and template strand separate, and nucleotides complementary to the template strand will be brought to the template and will be polymerized to form the RNA chain. And so the convention is, if there's only one strand there, it's written 5' to 3', and it's the coding strand. If there are two strands, they're written 5' to 3', and the top strand being the coding strand, and the bottom strand written 3' to 5', and that will be the template strand. The transcripts of many prokaryotic genes are polycistronic, and this means that there's more than one coding region in the messenger RNA. For instance, this one has 3 coding regions, and these coding regions are also called open reading frames. They are the regions that are going to be involved in the translation of the protein. For instance, coding region number 1 can give rise to protein number 1, coding region number 2 and 3 give rise to proteins number 2 and 3, for instance. And so this one messenger RNA can give rise to 3 different proteins that are translated from different regions on the messenger RNA. Eukaryotes, on the other hand, are monocistronic. Their messenger RNA has only one open reading frame per message. This is not to say that eukaryotic messenger RNA is not complex. In fact, just the opposite. Eukaryotes have very complex messenger RNAs that have untranslated regions, and they have exons and introns. Introns means intergenic regions. These are the regions that are going to be cleaved out and that will not give rise to any of the protein. The exons are regions that are actually used for translation, and the untranslated region, the UTR, is involved in translational regulation. Another difference between prokaryotes and eukaryotes is that eukaryotes have a 5' cap, and these are modified nucleotides that are added to the end of the transcript during transcription. This 5' cap is used as a site for the ribosome to eventually bind and begin translation, and is also necessary to help protect the end of the messenger RNA transcript. Same thing for the polyadenylate tail that's found at the 3' end. This is used to protect the 3' end of the transcript, and it's also used in the regulation of the transcript. When eukaryotic messenger RNA is first transcribed, it's transcribed as what's called a pre-mRNA, and this pre-mRNA has both the introns and the exons. So that's how it's initially synthesized. But during the process of transcription, there are factors that will cut out the introns and splice the exons together to form different types of messages from the same messenger RNA. So for instance, even though there are 3 exons that are shown here, not all of them will be used to translate the eventual protein. Some will be used and some will not be. And so you can have different final transcripts, different mature mRNAs, resulting from the same pre-mRNA transcript. And so eukaryotic messages are capped, tailed, and spliced. In this diagram is a representation of RNA polymerase that's shaded in yellow. Associated factors that help to position the RNA polymerase at the promoter site are shown in gray. We'll talk about those later on in the semester. Capping enzymes, splicing factors, and polyadenylation factors are all bound to the C-terminal domain of the RNA polymerase. This is a portion of the RNA polymerase itself. And so during transcription, the RNA is synthesized, and you can see a portion of it coming out of the RNA polymerase. As it's synthesized, the capping enzyme put a 5' cap on the messenger RNA. Another thing I'd like you to see is that there is an orange section in the middle of this messenger RNA, and this represents an intron that's going to be removed for the message as it's synthesized. And so more of the message is synthesized. The splicing factors come into play now, and they remove the introns from the mRNA and splice the exons together so that there is one mRNA that contains the necessary exons and does not contain any of the introns. And now it's the job of the polyadenylation factor to form a poly-A tail. And so the mature message is going to have a 5' cap, all the exons that it needs, but none of the introns, and it's going to have a 3' polyadenylate tail. And now this message is ready to be translated. Just to give you an idea of the complexity of this, here is a gene that has 3 different types of transcripts that are made from it. And these transcripts result in 3 different proteins that are made from the same gene through alternative splicing. In this diagram, the introns are shown as light blue, and the exons are numbered and shown in various shades of a darker blue. So the gene is on the top, the pre-mRNA is in the middle, and the 3 different transcripts that result from alternative splicing are shown below. As you can see, not every exon appears in the final transcript. In this way, we can have multiple forms of a protein being synthesized from the same gene. This is important when we want to have changes in a protein to alter its function or regulation in specific cell types or at specific times in development. Here are some transcription notes for you. Transcription has these characteristics. It requires a DNA template which is read in the 3' to 5' direction. The synthesized RNA is complementary to the template strand. Transcription does not require a primer. Transcription extends the chain in the 5' to 3' direction. During transcription, the free 3' hydroxyl of the preceding nucleotide attacks the alpha phosphate of the incoming nucleotide triphosphate. This reaction yields 1 molecule of pyrophosphate and an RNA chain that's extended by 1 nucleotide. Pyrophosphate is immediately hydrolyzed after it's produced to yield 2 molecules of inorganic phosphate. This reaction is catalyzed by a class of enzymes that are called RNA polymerases. RNA molecules serve many purposes, and we saw some of those today. They can act as genetic instructions for protein synthesis. We saw this with messenger RNA, mRNA. This is an RNA copy of the genetic information that's necessary to synthesize a protein. It serves as a template for protein synthesis. RNA can also act as regulatory molecules. Post-transcriptional genetic regulation occurs in both prokaryotes and eukaryotes through the action of small interfering RNAs called siRNAs and microRNAs, miRNAs. And we'll talk more about these later on in the semester. And it's most important to know the functions of messenger RNA, transfer RNA, small nuclear RNA particles, and also ribosomal RNAs. Those are the main types that you need to know right now. The other ones we're going to deal with later on in the semester. So, now that we have the message, how do we get that translated into protein? The message itself is written in 3-letter codes called codons. Each codon represents either an amino acid or a signal to stop translation. In this mRNA we see here in yellow that's bound to the ribosome, we see 3 codons that are being used. The tRNAs are shown in red and white. Each of the tRNAs carries its own amino acid that's typical for that particular mRNA, and that is encoded in the 3-letter codes that are in the mRNA. Each tRNA finds its particular codon on the messenger RNA through complementary base pairing between the messenger RNA codon and regions of the tRNA called the anticodon. In this diagram, you can see the first tRNA has an anticodon UAC, and its complement is AUG. This particular tRNA is bound to methionine, and this is abbreviated MET. And so, this first tRNA has methionine bound, and it finds its own particular codon on the messenger RNA and binds there through hydrogen bonding to the template. The next one in line is for serine, and the final one is for glutamine. And this goes on and on down the messenger template until there comes a signal that says stop. And we'll look at this a little more closely in just a minute. But these are the basics. Here we see a tRNA. This RNA is folded up into a specific form caused by hydrogen bonding that forms stem loop structures. One of these stem loop structures contains the anticodon, shown in red. At the top of the tRNA molecule, there is an amino acid attachment site, and this is where the amino acid is going to be bound. tRNA might not look very significant, But let me tell you what, it truly is. Without this small molecule and the enzymes that actually stick the right amino acid on the right tRNA, we wouldn't be able to decipher the code that's present in the mRNA. This is the translator right here. And so this tRNA allows us to go from the language of nucleic acids to the language of amino acids. This is the link between the two, the tRNA. And so by complementary binding of the anticodon to its particular codon on the message, the right amino acid gets to the right place in the peptide chain. This is very important. Translation is catalyzed by enzymes called ribosomes that contain RNA and protein. The RNA is shown in orange and yellow, and the protein is shown in blue. Ribosomes come in two parts, a small subunit and a large subunit, that fit together to enclose the mRNA. Ribosomes are old enzymes, and their catalytic portions are still RNA. And this is thought to harken back to the time when it was an RNA world, and RNAs were the major catalysts. Here we see translation happening. The amino acids are being brought to the template by the tRNAs, and a peptide chain is being formed. In this diagram, the tRNAs are described by dark blue, and associated proteins that are translation factors are shown in light blue. These proteins do such things as escort the tRNAs to the ribosome and actually move things forward so that an empty tRNA can leave from one side and a new tRNA containing an amino acid can enter from the other side. So let's talk about the genetic code. Each code word is 3 nucleotides long, and it identifies a specific amino acid or a stop signal. Each codon only encodes one amino acid or a stop. A code that reads "the big dog ran" is always the big dog ran, not the Heb Ibby, for instance. There's no placeholder. There's no punctuation between the code words. It's not X the, X big, and so on. There's no punctuation. The code is degenerate. Most amino acids are encoded by more than one codon. There are 64 codons to encode 20 amino acids and stop. So there has to be some amino acids that are encoded by more than one 3-letter codon. The number of codons representing each amino acid generally reflects the frequency of that amino acid's use in protein. For instance, methionine and tryptophan only have one codon that represents them. Here we see the genetic code. The first position is shown on the left, and the second position is shown on the top, and the third position is shown on the right. And so for AUG shown at the bottom of the screen, going 5' to 3' on the messenger RNA, we're going to start with that A. We're going to go to the first position in the codon usage chart, and we're going to find the A. And then we know that what we're looking for is going to be in this row that's represented by that A. Now we have to look for the second position, and that's highlighted in yellow. So we have the A and we have the U. And so it could be ILE or MET. We have to go to the third position, and this is highlighted in blue, and that's G. So we're looking for G, and here we go. We have methionine. So AUG encodes methionine. Now we can see here that many amino acids are encoded by more than one codon. Proline has many, serine has many, arginine has very many, and besides this, there are also codons that say stop. For instance, UAA, UAG, and UGA all say stop. So codons can encode either amino acids or they can encode the signal to stop. And this is an important thing to remember. The thing is, the universal code that we just saw is not entirely universal. There are some things that are encoded differently if you're talking about things that happen in the cytoplasm and things that happen in the mitochondrion. For instance, UGA in the standard code that we just saw encodes stop, but in the mitochondrion it encodes tryptophan. These are really specific to the mitochondria, and there are some organisms that will also follow their own strange codon usage. As scientists, it's important to know this, otherwise you can make mistakes. So just like with transcription, we need to know where to start and where to stop with translation. The start site is really easy, and it happens differently in prokaryotes and eukaryotes. In prokaryotes, there is a purine-rich region just downstream from the start site of translation where the ribosome binds. So the ribosome binds to this purine-rich region. It's about 10 nucleotides away from the start site. And then the first AUG is used. So remember when we talked about the polycistronic messages? Each of those open reading frames is going to have their own purine-rich region where the ribosome will bind, and then the ribosome will bind to that region and look for the first AUG and start translating from there. The first amino acid that's brought to the template by the tRNA in prokaryotes is called F-met. This is a formylated methionine. So that's also unusual with prokaryotes. Eukaryotes are very different. Eukaryotes, as you'll remember, have that 5' cap, and this is what the ribosome is going to bind to. It's going to bind to the cap and then start synthesis at the first AUG after that. Instead of having formylated methionine, eukaryotes just begin with a regular old methionine, and that's going to be the first amino acid to start translation with. Now these methionines, even though they are in the beginning, usually, they can be cleaved off afterwards. And so what about stopping? How is translation terminated? Well, we saw before we stop translation with a specific codon that says stop. There are factors that will bind to these particular stop codons that will help to terminate translation. And speaking of stopping, this is the end of the lecture. I hope you have a really great day.